Hello everyone, welcome back to another session of regeneration biology. So in today's class, we will try to learn about how importance of regeneration associated various factors, that is the gene, which includes a gene induction events or sometimes the early immune response, etc. The broad categorization will be the activators and repressors of the regeneration response. So in this lecture, we will be trying to cover what are the major events that is occurring and how and why these events contribute significantly for a successful regeneration. Okay, so if you look into an overview, we know for a retina regeneration point of view, so we will restrict this observation with regard to retina regeneration. Reason being, it is one of the complex tissue, and why and how a similar tissue in mammal, say Muller glia, do not respond effectively. So we will get to know those little bit more in detail. So hence we restrict this with retina itself. So Muller glia is the major component of the retina, which consists around 40% of the total number of cells, and they are one of the last retinal cell types that is born during the development, and they function to maintain the retinal homeostasis and integrity, because the neurons need lots of support from the glial cell, and hence the neurons are able to perform their higher order task. So in mammals, the Muller glia responds to the injury in various ways that can be protective or they can be detrimental to retinal function, but they are often limited in their ability to create a effective regenerative response. They indeed make some cell types, but they are not effective in bringing in a proper regenerative response. By contrast, in teleost fish such as zebrafish, we all know, we have seen how zebrafish responds to injury. The response of the Muller glia to the retinal injury involves reprogramming of the Muller glial cells, and it even imparts the retinal stem cell with unique characteristics of a pluripotent cell, and allows them to produce a proliferating population of progenitors that are capable of fixing all the damaged retinal cell types. And many recent studies have revealed several important mechanisms underlying Muller glia reprogramming and retina regeneration, and that will help the researchers to design strategies. How can we implement these discoveries of the zebrafish into a mammalian system? So if you look Muller glia, it has got a transformation phase. You can see the adult Muller glia after injury, it gets into a reprogrammed Muller glia. That means you have a color change now here in this picture. And it also have got interkinetic migration and asymmetric cell division. So this reprogrammed Muller glia is now getting into a proliferative phase, and often the hallmark is asymmetric. It do not have a symmetrical cell division. And these divided progenitors now expand in number, and it will form some columns of cells. In the retinal section, you can see it. They will form expand in number and the progenitor amplification. And this amplified progenitors, they will now give rise to all retinal cell types. So this is the overall theme of normal regeneration. So adult Muller glia and zebrafish responds to the retinal injury by reprogramming their genome, because all 6 pluripotency inducing factors are naturally induced in the Muller glia, and which allows them to get into a pluripotent stem cell status, to acquire stem cell properties. This reprogramming results in the interkinetic nuclear migration to the outer nuclear layer, because the damage has happened in all layers, and the Muller glia cell body resides in the inner nuclear layer. So unless these progenitors are migrating to various layers, you cannot you cannot fix the damaged cell types in different cell layers. So the cell division has to continue along with the migration, interkinetic migration. And this asymmetric cell division, which, like I told you, this reprogrammed Muller glia undergoes asymmetric cell division. Asymmetric cell division generates multipotent progenitors that transiently proliferates and restores the original Muller glia back. So it's not that there will be a short supply of the Muller glia because all of them are reprogramming. First of all of them do not reprogram. Even if the Muller glia reprogram, only a subset of cells that is derived from this reprogrammed Muller glia is undergoing the proliferation. And the multipotent progenitors migrate to all cell layers, exit the cell cycle when the regeneration purpose or the job of regeneration is completed, and regenerate all major retinal cell types. Now comes a important table. As you can see here, this is taken from a review published in Nature Reviews Neuroscience, and it's a huge table that depicts the factors affecting Muller glia reprogramming and proliferation. A lot of factors are listed here. Its function is listed here, and animal tested is listed here, and expression of it following an injury is listed here, and what is its effect, the role on the regenerative response. For example, delta notch is one of the first factor, and its function is basically transmembrane ligand and receptor. Both of them are present on the membrane, and it is been tested in animals such as bird, mice, or it's mentioned as rodent, fish, and all these species tried for retinal regeneration. They are induced soon after injury, and the effect can be quite unique in the sense it stimulates in birds and mice but inhibits in fish. See, one signaling pathway, both are induced, and you are seeing that in one case it is activated, in another case it is repressing. So we should understand the same signaling based on the species what you are studying is able to perform a different task. Same way you can say BMP is SMAD, and it's a secreted factor, and tested in rodent, and it is induced and it is stimulating. And you can see here another factor, that is DKK, which is a negative regulator of wind signaling. It's a secreted factor tested in fish, and it is suppressed. The DKK is suppressed soon after injury, and it is an inhibitor of regeneration. So the suppressor is blocked, then the regeneration is favored in zebrafish. So you can see, I will not go into each of this pathway. Those who are interested can read it. But what we should understand, there can be genes that are induced for a better cause in terms of regeneration, or there are some genes that are inhibited for a better cause of regeneration, and vice versa, other way around. So you can see some genes such as say TGF-beta is a secreted factor tested in rodent and fish, and it is suppressed, and it has been shown to inhibit. So in zebrafish, TGF-beta 1, some studies have shown to activate also, but in general it is an inhibitor. That means it is controlling, it acts like a brake in a vehicle. So this kind of regulation, in one hand you are pampering, in another hand you are punishing. So this is the way various factors act on. So the same table is continuing. You can see TNF alpha, you have got HSPD1. These are all different group of protein, family of protein that has been tried, and some of them are transcription factor, some of them are signal inducing factor, and some of them are transmembrane factor, and different species has been tried. A lot of them are tried in zebrafish simply because it is a well regenerating model. That is why lots of these pathways and factors are discovered in zebrafish. And you can see not all of them are induced, some of them are suppressed. LET7 microRNA we have discussed multiple times, that is a suppressed molecule, and LET7 if present it inhibits. So the inhibitors of regeneration are often suppressed, whereas the activators of regeneration are often induced in a well regenerating model. So the important question is, based on these factors, how do the Muller glia sense the injury? Someone has to tell the Muller glia that there is an injury. Of course, many factors contribute to it. Sometimes it may be reactive oxygen species, sometimes it may be the blood clot, sometimes it may be the PDGF, etc., that is induced because of a injury, a bloody wound. So these things contribute how the Muller glia is able to respond. However, what are the factors a Muller glia will be looking for? So secreted factors such as heparin binding epidermal growth factor HBEGF or tumor necrosis factor alpha and WINS, and they are and also ciliary neurotropic factor have been reported to contribute to injury induced Muller glia reprogramming and the progenitor formation. But some interesting factors are there. CNTF is not reported in zebrafish. It is normally present in mammals. CNTF gene is not reported in zebrafish. However, if you provide CNTF from mammal into zebrafish, it favors regeneration. So that is something interesting we should keep in mind. Other pro-inflammatory cytokines such as interleukin 6, in short known as IL-6 family cytokines, can contribute and supplement or complement for the absence of CNTF in the case of zebrafish. This doesn't mean that CNTF is non-functional in zebrafish. Only thing is the gene for CNTF is not present in zebrafish. Cytokines, probably IL-6 is taking that job, contribute to retinal regeneration in this species, that is zebrafish. Two factors that regulate Muller glia proliferation, that is ADP. You know, ATP, when it is degraded, it will become ADP, and it can further become AMP, adenosine diphosphate, adenosine monophosphate. Adenosine triphosphate is the energy source, and ADP can also act as a signaling molecule for the Muller glia proliferation, and TNF alpha are not only produced in the Muller glia but also appear to be released by the dying retinal neurons. So whenever there is an acute injury, these dying retinal neurons, they need not be glia, because retina has got lots of neurons also. So they die. When they are dead, it will release this ADP and TNF alpha that will stimulate the Muller glia. TNF alpha contributes to the injury-dependent induction of ASCL1A and STAT3, which are the two transcription factors. ASCL1 is a BHLH, basic helix loops helix transcription factor. STAT3 is a component of JAK-STAT signaling. STAT3 gets phosphorylated and gets into the nucleus and activate lots of genes, and whose expression is necessary for the generation of the Muller glia derived progenitors. So this picture, it kind of depicts this various signaling cascade that is contributing to the Muller glia reprogramming and progenitor proliferation in zebrafish. One point is the retinal regeneration requires the activation of a variety of signaling cascades, which I already told you. Not that every induced gene or induced pathway is pro-regeneration. Some of them will be anti also. But that doesn't mean that that anti-regenerative response or pathway is unwanted. It is needed. It's almost like telling I need a car without a brake. So this is what you should keep in your mind. Signaling pathways shown to regulate retinal regeneration are indicated in this picture by solid arrows, while those are indirectly implicated and hypothesized, which are shown in by dotted arrows, dashed arrows. And one factor, that is secreted factors. Normally some group of factors are there, like winds are there, and SFRP, many are there, which are, or sonic hedgehog, many are there, and this they are indicated present to be expressed outside the cell in this picture, and which can impact the Muller glia response or reprogramming. And arrows are pointing to top half of the nucleus that is present, that they stimulate and maintain positive, and the blunt arrow or blocked arrow indicates that is a negative regulator. So winds are secreted in a lipid modified fashion, glycoproteins, and they can bind to the frizzled family of receptors that will allow the stabilization of the beta-catenin. That is one of the pivotal pathways. We will see in the coming class also more about the Wnt signaling, and this allows that it will sequester disheveled from this degradosome complex, where normally the excess produced beta-catenin is degraded, and the dislodging of the disheveled, which is by a cytoplasmic phosphorylation acting downstream of this Wnt signaling events, that will stabilize. Because of this, the degradation of beta-catenin will be prevented. This will stabilize the beta-catenin, which will go to the nucleus and act onto the various target genes. And another factor, insulin and IGF, insulin like growth factor 1, they are secreted proteins that bind to the tyrosine kinase receptor that signal via IRS, that is insulin receptor signaling protein, that is called IRS, an adapter protein that couples the insulin and IGF receptor to PI3 kinase, that phosphatidylate inositide 3 kinase, and AKT. So this is the pathway in which the insulin and IGF act together. Another factor is HBEGF, that is heparin binding epidermal growth factor, is a transmembrane protein that undergoes ectodomain shedding, because it needs to be processed, because the enzymatic action causes an ectodomain shedding, and is a member of the EGF family of growth factor ligands, and it acts via epidermal growth factor receptors. Heparin binding epidermal growth factor acts via the EGF receptor. As you can see here, this is also taken from this review. You can see there are so many factors. There are membrane bound receptors act on HBGF is given here, insulin IGF is given here, and the DKK, which is a negative regulators of Wnt signaling, is given here, because these are all secreted factors outside the cell, and inside the cell this is the nucleus and this is the cytoplasm. Outside the nucleus is the cytoplasm, and this is the membrane. This is a nuclear region of the Muller glia itself. So the beta-catenin usually will be degraded in a steady state cell, but when Wnt signaling is on, the beta-catenin will be stabilized and it will go to the nucleus and creates lot of pro-mitotic gene that will be turned on. You can also see the Lin28 negatively regulating the LET7, which indeed influence the translatability of various regeneration associated genes. So you can see this picture in a nutshell. You can see there are both pro and anti-regenerative responding molecules combined together. In general, the reprogramming of the Muller glia depends on how effectively these pro and anti-regenerative events are triggered. Another factor is FGF, fibroblast growth factors. They are secreted molecule that bind to the fibroblast growth factor receptors. And EGF, epidermal growth factor, and FGF receptors are tyrosine kinase receptors that they can signal via MAP kinase, that is mitogen activated protein kinase, and ERK kinase, that is extracellular signal regulated kinase. This is the role in which the EGF and FGF is acting. And cytokines, which are mainly secreted by your immune cells, cytokines are secreted proteins that often signal through the receptors that lack their intrinsic tyrosine kinase activity. They don't do their job unless they bind to a ligand. Cytokine receptors are often coupled to JAK, that is also known as Janus kinase, and which is meant for the STAT3 activation. JAK proteins are non-receptor tyrosine kinase that transduce cytokine mediated signals by phosphorylating another protein called STAT. Signal transducers and activators of transcription. That is a STAT is the a short form of this long name to this protein. So the phosphorylated STAT can go to the nucleus and act onto the target genes. Then comes the TGF beta, that is transforming growth factor beta, is a secreted protein. They have a differential role in mammals. It is normally anti-proliferative, whereas in zebrafish it can be depending upon which TGF beta. TGF beta 1 is pro-proliferative, whereas TGF beta 3 is not pro-proliferative. Another microRNA molecule is LET7 microRNA, which we have discussed sufficiently, hence I will not go into the details, which is a negative regulator of reprogramming. So LET7 must go off if the reprogramming of the Muller glia has to happen. Tumor necrosis factor alpha, which is a secreted cytokine that acts via the TNF receptors that regulate the cell signaling and the gene expression. And delta notch signaling is another signaling event that has, both of them have got a single pass transmembrane protein, and they have to be present in the adjacent cells, and we often refer to as juxtacrine signaling. And then comes the last part, that is the ECM, extracellular matrix, can also signal the adjacent cell via the transmembrane integrin receptor to regulate the cell function. So from this we understand that the signaling events are not just initiated by triggered by one point. They are acted upon by a plethora of angles and directions onto a given cell. So let us quickly look into what are the major activators of a regenerative response in zebrafish. Retinal injury results in Wnt gene expression and the beta-catenin stabilization in the Muller glia derived progenitors. Inhibition of Wnt signaling, it can be done by multiple ways. You can use overexpress DKK, or you can knock down some of this gene, or create an knockdown of the beta-catenin itself. There are different ways of blocking Wnt signaling. Suppressors, and if you block the Wnt signaling, it can suppress the progenitor formation in the injured retina. So the pharmacological activation of the beta-catenin signaling pathway, that means you are stabilizing the beta-catenin, which is independent of the availability of the Wnt ligand. So Wnt ligand acts on the Frizzled receptor, that is a cell membrane associated job. Now you bypass the whole thing. You, no matter what is happening, you can stabilize the beta-catenin. That is by blocking one of the phosphorylation event which is necessary for the beta-catenin degradation. So the one of the phosphorylating molecule of the destruction complex is GSK3 beta, a glycogen synthase kinase 3 beta. So if you block it using an inhibitor, it can stimulate the Muller glia reprogramming and progenitor formation even without an injury. This is what you should keep in your mind. If you can stabilize the beta-catenin without an injury at all to the retina, it will start a regenerative response. These treatments can bypass an inhibitory retinal environment, and that results in part from a pan-retinal expression of the Wnt antagonist DKK. DKK is present throughout the retina. You don't want any unwanted proliferation happening in the retina. So soon after injury, in a normal regeneration event, the DKK will be downregulated. But when you are stabilizing the beta-catenin, you do not have to bother about the DKK at all. Let the DKK block the Frizzled receptor, doesn't matter. You are dealing with inside directly in the cell, and that too at the level of beta-catenin. So if you look into few more factors, CNTF, ciliary neurotropic factor, can stimulate Muller glia to generate progenitors in the uninjured zebrafish retina that suggests that the JAK-STAT signaling may also be involved in the Muller glia reprogramming and retina regeneration. So they also contribute, which is exclusively done by CNTF. Induced retinal injury can stimulate STAT3 expression in both quiescent Muller glia and also in the Muller glia derived progenitors. And the STAT3 knockdown, if you block the STAT3, it can inhibit the progenitor formation. So although other signaling events are perfectly happening, you cannot get a normal regenerative response if you get rid of one component. It's almost like assuming if there are 10 components needed to make a dish, if you got rid of one component, say salt, you put everything but you did not put the salt. So you cannot say I put everything but I just skipped the salt, but it will not taste correct. It will not be a servable dish. Same logic applies to individual signaling events. A Muller glial cell acquires the properties of the retinal stem cell, usually by the reprogramming of its genome to express genes that allow it to generate multipotent progenitors for the purpose of regeneration of the retina. So gene expression has to underlie before a Muller glia reprogrammed. And there are many negative regulators also there in the regenerative response. Plenty of negative regulators are there. Retina regeneration is driven by the activation of signaling pathways that stimulate Muller glia reprogramming and also progenitor formation because of these activators, and also suppression pathways that drive the Muller glia differentiation and quiescence. Sometimes some pathways, they are not wanting the Muller glia to continue to proliferate. They want the Muller glia to differentiate. So if the differentiation is happening in a prematured manner, then it is not. It's almost like, someone who is a 5-year-old person, you are expecting them to go for a job. It doesn't help because there is a maturation process, or there has to be a sufficient buildup of progenitors. So the gene expression has to continue for a fixed amount of time, and if it continues forever also, then there is a cancerous kind of response will be there, because you have only progenitor after progenitor after progenitor. You don't want that. So progenitor should come and then it should differentiate. So LET7, which we discussed many times, and another negative regulator, LET7 microRNA is a negative regulator of regenerative response, and DKK is another negative regulator, which is a LET7 is a RNA molecule, whereas DKK is a protein molecule. LET7 RNA when present, microRNA when present, it makes sure that there is no translation happens to those mRNAs which has got the LET7 binding site. And many of the regeneration associated genes have LET7 binding sites. Hence, you can expect even though they are induced, those genes are induced, they will never get translated. Same with the DKK. Even if Wnt ligand is present, unless the DKK goes off, the Wnt ligand simply will not be able to do any job onto the Frizzled receptor. and they are inhibitory pathways that help to maintain zebrafish Muller glia in a quiescent state. It means inactive state, which is necessary for a normal retina. TGF-beta signaling inhibitors, they can enhance the progenitor proliferation especially in the mammalian retina, and it can also do in zebrafish retina to some extent. And TGF-beta induced homeobox protein, that's called a TGIF1, and sign oculus homeobox 3b 6-3b, these proteins are involved in implementing the TGF-beta signaling, how effectively it is regulated during TGF-beta signaling. These inhibitory molecules or inhibitors are transcriptional co-repressors with multiple targets, and their effect on the TGF-beta signaling in the injured zebrafish retina remains still under exploration, because like I told you, TGF-beta 1 acts in a pro-proliferative manner in the early phases, whereas the same TGF-beta 1 acts as an anti-proliferative in the late phases, whereas TGF-beta 3 acts like an anti-proliferative molecule throughout the retina regeneration. So these are all some nuances one has to test. Notch signaling also appears to play an inhibitory role during zebrafish retina regeneration. So this one has to understand very clearly. It do not inhibit the proliferation in total, rather it inhibits the expansion of the zone. Say for example, there is an injury and you wanted injury response should be in 1 square centimeter area. I'm giving a very random figure, 1 square centimeter area, and you don't want in a 10 square centimeter area. But if the notch signaling is inhibited, instead of 1 square centimeter area, it becomes 10 square centimeter area. So if you want a responsive zone to be restricted in the required spot, then the notch signaling should be active. If you block it, it will become, it's almost like tying a dog to a leash. If the leash is 1 foot long, it will run around only 1 square foot area. But if it is 10 foot long, it will act on 10 square foot area. So same way, delta notch signaling restricts the zone of regenerative response. So that is why notch signaling also is considered as a negative regulator of however, unlike most inhibitory pathways which are suppressed following retinal injury, notch signaling components such as delta A, B, C, notch 1, notch target genes such as HER4 are induced by injury. So this is what you should, like I told you earlier, not every genes that are induced are favoring regeneration. Sometimes some active signaling is not favoring regeneration. In case of delta notch signaling, these ligands and the receptor, delta is the ligand and notch is the receptor, they are induced mainly to restrict the zone of regeneration in the case of retina regeneration. So in this picture, you can see how different gene expression events, they congregate together, and where the central focus is given to ASCL1A, which is a basic helix loops helix protein. It impacts the Muller glia reprogramming and proliferation by regulating Wnt signaling pathway, but it can inhibit the DKK. Being a transcriptional activator, it can inhibit the DKK, and it is indeed necessary for the action of the Wnt protein onto their target. But how ASCL1A is doing this? Because ASCL1 is a transcriptional activator. So how ASCL1A is doing the negative regulation on DKK is through the activation of a repressor called INSM1A. So ASCL1A regulates the expression of insulinoma associated 1A, short form known as INSM1A, which is a transcriptional repressor that affects both Muller glia reprogramming and progenitor cell cycle exit. So it acts in two ways. INSM1A can block its own expression, INSM1A can block on DKK expression, and INSM1A can block various other factors. So those are listed here. ASCL1A acting on various signaling and growth factor response to it, and how ASCL1A is influencing different factors are listed in this graph. We will study about the regeneration response in the next class in more in detail. Thank you.